Hello. I'm going to talk about comparative phylogenetics of the ICEHin1056 family and insights it provides us into the evolution of mobile genetic elements. Perhaps in direct contrast to the slightly obscure title, this has direct relevance to human health, and the reason is antibiotic resistance. Described as a threat as big as terrorism by the Chief Medical Officer of the UK, Dame Sally Davis, and as causing 2 million episodes of disease and more than 23,000 deaths per annum by CDC in Atlanta, antibiotic resistance has been hitting the headlines as never before. Since the seminal work of Lederberg and colleagues showing that plasmids were key for transferring genes laterally, that is, other than from parent to offspring in bacteria, we have known that the lateral gene pool is very important. ICEs, integrating and conjugating elements, are relative newcomers to the mobile gene pool. However, it's increasingly being recognized that they are very important in transferable antibiotic resistance. And today I'm going to talk a bit specifically about this one family of ICEs, ICEHin1056, and their contribution to the lateral gene pool, the presence of accessory genes on many ICEs in the family, and how they contribute to environmental adaptation. The concept of the usually accepted modular mosaic model for the evolution of mobile genetic elements, and how some data that I will show you appears to subvert that. The critical role of integrase genes in the behavior of mobile genetic elements, specifically ICEs, and some conclusions and future research. The Ice Age was a term coined by Vincent Burris, I think at the Pasteur at the time, when they were looking at the behavior of ICESXT, which is a family of ICEs present in Vibrio cholerae and responsible for the multi-antibiotic resistance seen in the 7th cholera pandemic. ICEs are made up of readily identifiable modules encoding maintenance, conjugation, and integration into the genome, and they are usually considered to be made up of mosaics, modular mosaics, where the modules evolve independently of one another. Phylogenetic studies of mobile genetic elements are increasingly being done and can provide us with interesting insights into the evolution of virulence and antibiotic resistance in bacterial populations. However, what I'm going to present today is novel in that it is the largest complete study so far of whole ICE elements. So there's a lot of very interesting work coming out of Eduardo Rocha's group looking at the phylogeny of integrase genes and type 4 secretion systems. But other than a small study on ICESXT, nothing looking at the phylogeny of whole ICE elements. My way into all of this is through Haemophilus influenzae and ICEHin1056. Haemophilus is a fastidious gram-negative bacterium, which is a member of the Pasteurulaceae. And since the introduction of the phenomenally successful Hib vaccine, it's been largely considered a medically solved problem. However, in the field of antibiotic resistance, Haemophilus influenzae has never really gone away. Non-typable Haemophilus, that is without a capsule, causes many diseases, including principally otitis media and exacerbations of chronic bronchitis, as well as bacteremias at the extremes of age. The respiratory tract infections caused by Haemophilus account for a huge proportion of the primary care consultations in the developed world, and correspondingly a lot of antibiotic prescription. It has been calculated that nearly 80% of consultations in the UK for otitis media result in a prescription for antibiotics, despite limited evidence of efficacy, and therefore resistance in the organisms causing these diseases drives further broad-spectrum antibiotic resistance, with the ongoing downward spiral of further broad-spectrum prescription and for further resistance. ICEHin1056 was first discovered in a Hib isolate causing a family outbreak of multi-drug resistant disease in the 1990s. And work by my colleagues at Oxford has shown that actually ICEs of this family are widespread in Haemophili and in fact account for over 90% of the antibiotic resistance seen in Haemophilus in a study of Oxfordshire children. This is a schematic diagram of the ICE, and you can here see the typical modules. At this end, we have the module which is usually annotated as being responsible for maintenance. Replication, autonomous of the bacterial chromosome, is not usually thought of being as a property of an ICE. However, ICEHin1056 can exist in a closed circular extrachromosomal form and therefore does not require the bacterial chromosome to replicate. And at least one other ICE, that in Bacillus subtilis, has also been shown to replicate independently. It is interesting that in some of the more recent work looking at the phylogeny of the type 4 secretion system, which I'll mention in a moment, that Eduardo Rocha's group have suggested that ICEs and plasmids as a distinction is artificial anyway, and they should perhaps be considered as interchangeable elements. Here, the type 4 secretion system encodes everything needed for conjugation, a pilus, and all the machinery required for getting single-stranded DNA from a donor to a recipient. And here at the end, we have the final core module, which is for integration, encoding a relaxase, which nicks and unwinds DNA, allowing single-stranded DNA to be transferred through the sex pilus, and an integrase gene, which I'll return to in a minute. ICEHin1056 also contains these two additions of accessory genes, a partial transposon 3, which encodes the beta-lactamase gene responsible for amoxicillin resistance, and a TN10-like structure encoding chloramphenicol and tetracycline resistance. All of this sounds very interesting and all very narrow and restricted to a single pathogen. However, a much broader picture was revealed by some early work by Derek Crook and Zaini Modzane in Oxford, who used some of the early genome sequences available to discover that the ICE had very close relatives in a number of other bacteria, including in Pseudomonas. Clearly, in the last few years, there's been an explosion in the amount of whole genomes available in the NCBI database, and the starting point of my study was to interrogate the NCBI database to see if other members of this ICE family could be found, how widely they were distributed, and whether their phylogeny could tell us anything about how the ICE was evolving. In order to find the ICE family, a core genome consisting of the concatenated amino acids of the 23 core genes of ICEHin1056 were used as a query sequence in a BLAST search using the tBLASTX algorithm. All the BLAST hits were aligned against the ICEHin1056 core genome in the Artemis comparison tool, and all the hits which showed conserved, all three modules were conserved, were then imported into the BIGS database, which was set up for me by Xavier Didelot, who I think is sitting at the back there, which allowed me to automate the identification and concatenation of the core gene homologs of the ICEHin1056 family. The results are very exciting. So out of more than 430 BLAST hits suggestive of related ICEs, I found 110 conserved syntenic elements. They were found across the beta and gamma proteobacteria, with a single representative in the alpha proteobacteria. All the ICEs identified as family members had at least 20 of the 23 core genes, and the habitats they were found in are enormously divergent, from the throat, obviously in the case of Haemophilus, to the silt of soda lakes in the case of a strange bug called Thioalcalovibrio, which is an extremophile and lives happily here alongside the flamingos. The ICEs in the family are enormously diverse in all properties except their core genome. They range in size from 20 to 250 kilobases. And you can see here on this schematic diagram that they have accessory genes inserted at one or more hotspots. This is the schematic of the core genome. All the ICEs have an insertion at least at one hotspot, though none of them have them at all of them. And the accessory genes inserted into the ICEs are as diverse as the hosts in which they're found. They range from the antibiotic resistance genes in Haemophilus influenzae to the virulence factor, the VI antigen in Salmonella typhi, through to enzymes which degrade toxic metals such as mercury and arsenic in organisms found in the slag heaps of toxic metal mines. What they all have in common is that they confer on the host organism environmental niche adaptation, supporting the theory that mobile genetic elements provide an excellent platform for acquisition of accessory genes which allow the organism to increase its environmental niche at little cost to itself, as the inserted genes will not be going into a core host gene. This is a picture of showing you the conserved synteny of the elements using an Artemis comparison tool plot, and actually I have borrowed this from Zaini Modzane. What you can see here is the three core modules are conserved with high levels of homology, but between the modules, and in some cases within them, we see large insertions of accessory genes. In order to generate the phylogenetic trees of the core ICE, all the ICEs were concatenated and then the sequences aligned in muscle, and a maximum likelihood phylogeny derived using the program PhyML. The results are seen here in this tree, and I accept this is rather busy and probably difficult to see from the back. But what is striking is that this is essentially a tree of two halves. So in this half, we have a number of ICEs, all of which are relatively divergent from one another, and which cluster with the taxonomy of their host. So here in pale blue, we have the ICEs found in the Pastoralaceae, including Haemophilus. In the purple, ICEs found in the Enterobacteriaceae, and here, the Pseudomonads. These are all ices which have GC% contents which are the same as those of their host genomes, suggesting they have been in the host genome a long time, and their divergence from one another suggests they are relatively fixed within their taxonomic grouping. At the bottom of the tree, however, we have a completely different picture. Here, in this cluster, we have ices which are much more closely related to one another than their host organisms are to each other. We have here the single alpha proteobacterium called Parvibaculum, and then a very mixed picture of beta and gamma proteobacteria, all of whose ices are very closely related at the nucleotide and amino acid level, suggestive of much more recent transfer. When seeking a reason for why we should have a tree that looks so different in its top and bottom halves, two things are striking. Firstly, that largely at the top, these are organisms which have limited habitats. Clearly, the Pasturulaceae are very limited, usually to a single host type, whereas these organisms at the bottom are environmental type organisms, occasionally found as pathogens, things like Pseudomonas burkholderia. Nonetheless, they are environmental organisms which enter the human system only as opportunists, and they are likely to live together in very diverse communities. The other thing that's very different between them is the integrase. And this comes on to the final part of the story of the ices. I showed you the picture at the beginning that the ices all have an integrase at the far right-hand end. There are two integrase lineages found in the ices of this family. They're either a ZER, which is closely related to the recombinase, which is responsible for correctly partitioning bacterial chromosomes at cell division, or a P4, prophage-like integrase. And the identity of the integrase present on the ice governs the behavior of that ice. Both integrases ensure that the ice inserts sites specifically, though the sites are different. Ices with a ZER-type integrase conjugate at a much higher rate in vitro than those do with a P4. And actually, previous studies have suggested that the presence of a P4 integrase on an ice or genomic island is actually a marker of fixity in the genome because the conjugation rate in vitro is so low. However, I would suggest that the data I showed you on the tree preceding this slide suggests that actually the converse is true. And even if in vitro the conjugation rate is very low, there is promiscuous spread of P4 integrase containing ices between many different species and genera. And when we recolor the tree by integrase lineage, this is what you see. In yellow, these are ices which contain a P4 integrase, and they contain all the ones that are more closely related to each other than to their host taxon, and all of these go into a tRNA glycine gene. In shades of blue and green, we have the ices which have a ZER-type recombinase, and again, their coloring denotes both the ZER lineage and also the tRNA insertion site specificity. These go into a leucine, these into a phenylalanine, and these into a lysine. So to sum up what I've said, I believe that this study, which is the largest study of complete mobile genetic elements, specifically ices to date, suggests that there is powerful selective pressure on the core ice as an entire unit. The conserved synteny seen in these ices argues against the model of modular mosaicism. It suggests that the integrase is a key determinant of intertaxon transfer and the ability to spread beyond the narrow taxonomic grouping in which the ice is found, and that the spread within the broader taxonomic communities is consistent with the concept of a supergenome and genetic exchange communities within very broad taxonomic groupings. Understanding the dynamics of gene flow in these kind of environments could help us understand better the emergence and spread of virulence factors and antibiotic resistance relevant to human and animal health, and at least potentially by understanding them to try and limit these exchanges. Further work to be done on this ice includes understanding the regulators of transcription and transfer and delineating the relationship, which is like that between a host and a parasite, between the bacterium and its mobile genetic element. I'd like to thank all the people in Oxford who worked with me, specifically my supervisors, Derek and Derek, Xavier for help with the phylogenetics, the Wellcome Trust who funded this, and of course the new Division of Microbiology and Infection for giving me the opportunity to carry on with such exciting research.